The overall goal of this technique is to make, calibrate, and use monopolar potassium ion selective microelectrodes. Using such electrodes enables the quantitative assessment of evoked potassium ion concentration dynamics in adult brain slices.
This method can help answer key questions in physiology, such as identifying the cellular and molecular mechanisms for potassium ion homeostasis in the central nervous system. The main advantage of this technique is that in addition to being an easy and straightforward method of fabrication, microelectrode sensors represent the gold standard for the measurement of potassium ion concentrations.
To prepare the borosilicate glass capillaries for selenization, place them in a 50 milliliter conical tube. Fill the conical tube with 1 molar hydrochloric acid and incubate the glass capillaries in hydrochloric acid with gentle agitation overnight for a minimum of 6 hours. Afterward, briefly rinse the capillaries with 70% ethanol and then dry them completely at 100 to 120 degrees Celsius for 6 to 8 hours. Store the washed capillaries in containers with anhydrous calcium sulfate desiccant for up to 4 weeks before further use. Prior to selenization, pull the capillaries to a fine tip using a microelectrode puller. Then, place the microelectrodes in a glass container using autoclavable tape so that the electrodes are elevated from the bottom to prevent tip breakage. Next, remove approximately 0.5 milliliters of 5% DDS selenization solution from its container using the nitrogen replacement method.